Hi everyone, I'm going to be talking to you today about mass transport in animals, which is the next topic that we're going to be studying together. So what you can see in front of you here is the specification point. So let's just take a minute to have a look at what we're going to be learning about. Our lessons are not going to be following the exact order that you see here. So later on we can see we're going to be learning lots about hemoglobin. We're going to learn about what they are, what they do, and different types of hemoglobin. We're going to be learning about the general pattern of blood circulation in a mammal, which we'll be starting today. But the main thing we're going to be doing in today's lesson is looking at the structure of arteries, arterioles, and veins in relation to their function, and the structure of capillaries and the importance of capillary beds as exchange surfaces. And we might, if we've got time, get onto the formation of tissue fluid. So we're going to be focusing on these things here, these three paragraphs that I have highlighted. And the hemoglobin and the heart we will get onto at a later date. So let's just take a minute to have a look at this paramecium. This paramecium is a single-celled eukaryotic organism. It's a protist and it moves around in fresh water. We can see it's we can kind of see here it's all covered in little cilia, these tiny little hairs that waft about to move it around. We can also tell it's eukaryotic because it has these internally bound vesicles, so it's got internal structures that are complex. So when you've got very small organisms like a paramecium, it has got a really large surface area to volume ratio. And because it's got a really large surface area to volume ratio, it doesn't require a complex mass transport system because the surface area to volume ratio is large enough to suit its metabolic demands. So oxygen can come in, carbon dioxide can come out, it can lose other waste products just directly across its surface. It doesn't need a complex mass transport system because there is a short diffusion distance between the outside world and the center of the cell. Whereas in more complex organisms, like mammals, we are big, we are multicellular. Our surface area to volume ratio is too small that molecules from the outside that we need to get in would have too far to travel, and so we need to develop a complex transport system. The type of transport system that exists depends on the metabolic demands of that organism and its size, so how complex that it is. So we've got a trade-off between efficiency and complexity. So in insects we have a very simple system, but the problem with that simple system is that gas exchange and nutrient exchange tends to be relatively low. And so you've got this simpler system which is easy but doesn't allow you to be very metabolically active, compared to a much more energy consuming, hard to maintain closed system that we see in fish and mammals and reptiles, but which allows them to be more metabolically active. So let's just take a look here at our diagram of the locust. So we can see that locusts have an open circulatory system. So all arthropods, including insects, crustacea, and most mollusks, have an open circulatory system. Essentially what that means is that the blood is not enclosed in blood vessels. Insects don't have arteries, veins, and capillaries. If you were to cut a tiny insect open, you wouldn't see a tiny little four-chambered heart, you wouldn't see a tiny aorta, you wouldn't see anything like that. There's just a big cavity in the middle of the body, which is called the hemocoel. And the fluid moves around within that hemocoel. So the fluid is actually referred to as hemolymph. This is because the fluid is actually a mixture of both blood and interstitial fluid, which is like tissue fluid, so the fluid that bathes all of the cells and organs. So as the extremely simple heart beats and the animal moves, this causes the hemolymph to circulate around the organs within the body cavity within the hemocoel. It then is able to re-enter the heart and be pushed around again. It re-enters the heart because there are tiny holes in the heart called ostia. So this movement allows for exchange of gases and exchange of nutrients. So like we said before, the exchange actually tends to be relatively low, but it's a trade-off because it's very simple. Then as we move up in complexity, we head to the closed circulatory system in fish. So this closed circulatory system is the first time we see actual vessels. So now our blood is trapped in vessels, arteries, veins, and capillaries. This diagram is a little bit better for seeing how it looks, this single loop circulatory system in fishes. So fish have a single circuit for blood flow and a two-chambered heart. that has only a single atrium and a single ventricle. So the atrium collects blood that has returned from the body, and then the ventricle pumps that blood to the gills, where gas exchange occurs and the blood is reoxygenated. This is called gill circulation. The blood then continues to flow through the rest of the body before arriving back at the atrium. This is called systemic circulation. The unidirectional flow of the blood produces a gradient of oxygenated to deoxygenated blood around the fish's system. This essentially results in a limit in the amount of oxygen that can reach some of the organs and tissues in the body, because by the time it gets there, it might have run out. This reduces the overall metabolic capacity of fish. This is the problem with a single system. By the time the blood reaches some organs within the body, it may well have run out. This imposes a metabolic limit on fish. This means that fish cannot be as active as other animals. So, because fish are less metabolically active than other animals, their circulatory system is sufficient for their metabolic needs. Whereas when we start to think about more metabolically active animals like mammals, we then need a more complex system, and that's why we have a double circulatory system. But before we get to mammals, let's take a look here. So the double circulatory system is where blood travels in two directions. So it goes in two circuits, one through the lungs and back to the heart, which is called our pulmonary circulation, and the other through the rest of the body, the rest of its organs, including the brain, which is known as our systemic circulation. Amphibians don't actually have to rely solely on their circulatory system. Gas exchange also occurs through their skin during pulmonary circulation and is referred to as pulmocutaneous circulation, but please do not worry about that because you do not have to know about that. So let's just think about amphibians and some reptiles first. So amphibians and some reptiles have a 3-chambered heart as opposed to the 2-chambered heart we saw in fish. And we can see that in this 3-chambered heart there are 2 atria and there is 1 ventricle. So the 2 atria, we can see they receive blood from the 2 different circuits. So the atria receive blood from the pulmonary circulation, and they also receive blood from the systemic circulation. Then the blood travels into the single ventricle, which is this one down here at the bottom. When it gets there, there is some mixing of the blood in the heart's ventricle. So essentially, oxygenated blood that's come from the pulmonary circulation mixes with the deoxygenated blood that came from the systemic circulation, and this adds a level of inefficiency. It reduces the efficiency of oxygenation. This essentially means that when blood is pumped out to the systemic circulation, it is made up of a mixture of both oxygenated and deoxygenated blood. Now, this is advantageous because the arrangement like this means that the high pressure in the vessels pushes the blood to the lungs and the body. So a high pressure can be generated by the heart, right? Whereas in the fish, you couldn't generate a high pressure because you couldn't have high pressured blood traveling through the gills because the gills have these tiny, tiny fragile capillaries. And so the blood has to always travel at low pressure. But because in the amphibians, the pulmonary and the systemic circulation are kept separate, the high pressure generated by the ventricle that travels to the systemic circulation doesn't affect or risk damaging the capillaries surrounding the lungs. So one advantage is it means that they can generate high pressure and push the blood to both the lungs and the body. And the mixing is a little bit mitigated by this little ridge. So there's a little ridge that slightly separates it out. But the disadvantage, of course, is that their organs and tissues receive blood that is not as well oxygenated as it could be. So this, we can sort of see quite clearly how this partial septum could keep developing until we end up with the heart that we know and love, that we find in mammals, birds, and crocodiles. So in birds and mammals, the heart is divided completely into 4 chambers. Even though it says crocodilians as well, the crocodile's heart is slightly different to ours, so we're not really going to focus on it too much. We're just going to focus on birds and mammals. So the heart is divided completely into 4 chambers that you already know. You learnt about it at GCSE. You've got the 2 atria, the 2 ventricles, and oxygenated blood is fully separated from deoxygenated blood. This creates a really efficient system. So it enables the the body enables birds and mammals to live the life they do. It supports this homeothermic lifestyle, so we are able to maintain our own internal body temperature. Mammals can be active both at daytime and in the night time. So we have really high metabolisms. We generate a lot of heat. We need a really efficient circulatory system to have this high respiratory rate in order to do that. Like, we have a really high respiratory rate compared to other animals. Like, compare us to things like snakes, right? Snakes might only need to eat like once a week or once a month, depending on the size of the snakes, whereas I need to eat at least 6 times a day. And this is because I am a little furnace. We're generating loads and loads of heat all the time, and this is really expensive. So we need to be able to get the nutrients to our cells. We need to be able to get the waste products away from our cells really efficiently. So sometimes when we look at a diagram like this, it's like, oh yes, I can see this evolution, dum-de-dum-de-dum, off we went in this direction, fish, amphibians, reptiles, birds, mammals, blah, blah, blah. It makes total sense. But actually, and this is really important, the 4-chambered hearts of birds and mammals evolved totally independently from a 3-chambered heart. When we look at structures which look really similar, and we think that they're very closely related, but they didn't. They evolved totally independently. So two things evolved totally independently to solve the same problem, and now they look the same. We call this convergent evolution. So this is an example of convergent evolution. So for the rest of this video, we're going to be focusing on the mammalian circulatory system. So when we think about the mammalian circulatory system, we know that there are 2 systems that we are thinking about. There is the cardiovascular system, i.e. our blood vessels and our heart, and there is the lymphatic system. The lymphatic system carries lymph around our body. The lymphatic system is sort of like the slightly less famous, lesser known little sister of the cardiovascular system. Everybody knows about blood vessels. Who knows about lymphatic vessels? Not so many, but they're incredibly important. So the lymphatic system is a network of tissues and organs that mainly helps to rid the body of toxins and other waste products. The primary function is to transport lymph. Lymph is a fluid which contains white blood cells throughout the body. So our lymphatic system primarily consists of lymphatic vessels, which are quite similar to veins and capillaries in the circulatory system, but they are open-ended. So these are open-ended ducts. So can you kind of see how they just sort of end? Like, it isn't a closed system, it's an open system, and it ends in the tissues of our organs. So later on when we talk about the formation and the draining of tissue fluid, these lymphatic vessels are going to become really important. You also have loads of lymph nodes, and you can kind of see them here on the diagram. We've actually got hundreds of lymph nodes in our body. We find some around the lungs, we find some around the heart, we find some in our groin and in our neck. The largest lymphatic organ is our spleen. Your spleen is kind of one of those organs that people are like, oh, I'm not sure what the spleen does. The spleen filters our blood. So the spleen controls the amount of red blood cells, and it helps to fight infection. So the spleen is a really important organ. So it detects potentially dangerous bacteria and viruses and other microorganisms, and lymphocytes can be produced, which will then protect us by producing antibodies and killing any foreign microorganisms. So lymph is a really important part of our immune system. And this is why our lymph nodes will often swell when we're sick. Sometimes when you're sick, your mum or your dad might like look at your neck and be like, oh yeah, your neck looks a bit swollen. They're looking at your lymph nodes and they're saying, oh, they're swollen. They're full of white blood cells. Whereas if we look over here at the cardiovascular system, we can see how this system of arteries, veins, capillaries runs throughout your body. I'm just going to show you next a few absolutely stunning images from a guy called Gunther von Hagens. I don't know if you've ever heard him. He's well worth Googling, but he developed a technique called plastination, and through plastination you can essentially preserve a particular body part while also getting rid of all the rest of the tissue surrounding it. So what he did in one of his exhibits was he preserved all of the blood vessels in different organisms, just to see how many blood vessels we actually have, and it's amazing. So take a look here. Isn't this absolutely incredible? So this is a human heart, and we can see how in this heart only the blood vessels have been preserved. So we can see that the heart, we can see this beautiful aorta up here at the top, and then we can see all of these coronary arteries running the outside, and then we see millions and millions of capillaries. And look at this one, this is absolutely incredible. So this is a horse's head, and we can see all of the fine capillaries just everywhere, just reaching everywhere. And in your body, there's no cell that is more than 2 cells away from a capillary. That is a really good way of imagining just how dense capillary networks are in your body. And I just love this one as well. So in this one you can see the shark, and it almost looks furry. You can just see how the capillaries are everywhere. So let's just take a minute to look at this really simple layout of the circulatory system. So here we can see our heart, and we can see the heart. You've got the vena cava, you've got the inferior vena cava coming from below the heart, and the superior vena cava coming from above the heart, bringing deoxygenated blood back to the right atrium, down into the right ventricle, and then it will go up via the pulmonary artery to the lungs, where it will become oxygenated. And once it's become oxygenated, it will then travel from the lungs back to the heart via the pulmonary vein, into the left atrium, left ventricle, and then up out of the aorta and off to the rest of the body. And then the aorta splits, so it can travel up to the arms and the head. And we can see the carotid artery here, the subclavian artery, and then it goes down to the liver, down to the stomach and the intestines, down to the kidneys, the legs, and so on and so on. And then once it leaves those organs, it has been deoxygenated, and then it will travel back to the vena cava. Now, the only blood vessel in your body that travels from organ to organ and not to or from the heart is the hepatic portal vein. The hepatic portal vein is a really important one, because when you eat food and you absorb all of those nutrients and things from the food into your bloodstream in the stomach and in the intestines, you might have absorbed something bad. You might have absorbed some toxins. Maybe you drank some alcohol. Maybe you ate some poison. You don't want that running all the way through your body. You need a protective mechanism, and that is the purpose of the hepatic portal vein. It takes this blood to the liver, and we know the purpose, one of the main functions of the liver, and it's got lots of functions, but one of the main functions is to detoxify the blood. So anything bad that you've eaten or drunk will go to the liver first to be detoxified, and then once we're sure our blood's a bit safer, we send it around the rest of the body. Sometimes we can take our knowledge of the circulatory system for granted. We can be like, oh yeah, this is obvious. If somebody didn't tell me this, I'd probably figure it out for myself, right? It seems so obvious, but that's simply not true. When we learn these things, we are standing on the shoulders of giants. It's really important that we understand that everything we learn in biology, even things that seem very simple, we learn them because somebody, not just somebody actually, hundreds of people have devoted thousands of hours or their entire careers, their entire lives to learning these things. There's so much passion behind every fact or every snippet of information that we get to learn. So I was going to talk to you about these two men. The first one is Galen and the second one is William Harvey, and we'll talk about each one in turn. So Galen is from the 2nd century, and he was a Roman doctor who gained knowledge from animal dissections. So he understood the importance of the pulse, and he understood that blood was moving around our body. But he made quite a large number of mistakes. For example, he thought that the liver made blood for the veins and the heart made blood for the arteries, which we now know is wrong. According to the gallenic system, blood is created in the liver from ingested food and flows to the right side of the heart. Some of it flows to the lungs, where it gives off sooty vapours, and some flows through invisible pores into the left side of the heart, where it gains vital spirits. When mixed with pneuma brought in from the trachea, several arteries flow into a fictional rete mirabile at the base of the brain, where vital spirits are changed to animal spirits before being distributed to the rest of the body through hollow tubes called nerves, where the blood is consumed by the tissues. So now we can look at that and we can be like, that makes literally no sense. But this was huge. This was what people thought and understood about the circulatory system for the next 1,500 years. So all all subsequent practices of medicine were built around this defective understanding of physiology for a really, really long time. And it was taboo to say anything against it, which is why that makes the work of William Harvey So important, because he was like a dog with a bone. He was one of the first people who was like, this makes no sense. This is what it's like. He is the first person that really understood the circulatory system for what it is and for how we learn it today. So let's talk a bit about William Harvey. So William Harvey was an English physician who was the first to describe accurately how blood was pumped around the body by the heart. So William Harvey was born in Folkestone in Kent, and his father was a merchant. He was educated at King's College in Canterbury and then at Cambridge University, and then he studied medicine at Padua in Italy. So he did a lot of dissection of animals, a lot. He did a lot of vivisection of animals. So he would cut dogs open while they were still alive, so you could see the pulse in their arteries. He first revealed his findings at the College of Physicians in 1616, and in 1628 he published his theories in a book, An Anatomical Study of the Motion of the Heart and of the Blood in Animals, where he explained how the heart propelled the blood in a circular course through the body. His discovery was received with great interest in England, but it was received with quite a lot of skepticism. He worked his whole life, and after he died, people were still struggling to understand it. And we're really lucky that now we know really how it works. And that all came from his research, so that he was really the starting point from us getting to understand the circulatory systems. He's really important. So let's take a look here. We're going to be talking about the structure of arteries, veins, and capillaries. So we know just generally that arteries carry blood away from the heart and into arterioles. Arterioles are smaller than arteries and control blood flow from the arteries to the capillaries. So blood is flowing at very high pressure in the arteries. If it was to go directly from arteries to capillaries, that would cause damage to the capillaries. So it's important that we have these sort of intermediate vessels where we can have a slight slowdown in the pressure. And then capillaries are tiny vessels that run through tissues and organs. The blood pressure is very low in the capillaries. Blood is also flowing at a very low speed, so we've got low velocity in the capillaries. This enables time for exchange of materials with our cells. And they link arterioles to venules. Venules are just the link organs between capillaries and veins, and then into veins, and then veins carry blood from the capillaries back to the heart. Let's just look here at the structure of arteries and veins. So we can see that they all contain the same basic structures. They both have endothelium, which is the single layer of endothelial cells that line the interior surface of blood vessels and of lymphatic vessels. Just looking at that word, endo means inside. And then we've got the basal lamina. The basal lamina is sometimes referred to as connective tissue. It's basically just like a whole load of glycoproteins and collagens and a bunch of stuff that essentially sticks the endothelium to the next layer, which is the elastic layer. The elastic layer allows the arteries to expand and relax. They expand and they recoil. This means they change their size as the blood is coming through with the pumping of the heart. So the heart is pumping, and the change in the size of the arteries due to the elastic tissue allows them to maintain a relatively constant pressure despite the pumping. So even though the pumping is bringing blood in these waves, the pressure remains the same because the size is getting bigger or smaller depending on the pressure change. So the elastic tissue has stretch and recoil, allowing the blood vessel, or the lumen, the space in the center of the blood vessel, to expand and then return to its original size. Outside the elastic layer, we have got smooth muscle. The smooth muscle allows them to withstand the high pressures generated by the left ventricle of the heart. At GCSE, it would have been fine for you to say to withstand the high pressures from the heart, but now we need to be a bit more specific, right? From the cardiac muscle in the left ventricle of the heart is the thing that is generating this force, this high pressure, and it is the smooth muscle and the elastic tissues that are allowing them both to withstand it, and the elastic tissue allows it to maintain a relatively constant pressure within the arteries. So if we look at the veins, we can see the veins have also got an endothelium. They've also got the basal lamina. They've got a little bit of smooth muscle, and then they've got this outer connective tissue. The outer connective tissue in both arteries and veins allows them to attached to other things in the body, so other organs and other tissues, allows them to be held in place. But they don't need the elastic tissue. This is because they don't need the stretch and recoil, because they're not being affected by the pumping of the left ventricle. The other thing that we see that's different in veins is the presence of valves. So because veins operate under low pressure, some veins have valves to prevent backflow of blood. So these valves will snap shut to prevent blood from flowing backwards. Okay, so let's take a minute to have a look at this graph. We can see this is a really important graph. There's loads of info on it. So we can see on the x-axis, we've got all the different types of blood vessels: arteries, large ones, small ones, arterioles, capillaries, venules, veins. And then we've got low to high on the y-axis and all the different things: low to high blood pressure, low to high velocity, low to high total area, all these different things. So let's start talking first about velocity. So we can see that blood is traveling at a high velocity in the arteries. Then when we get to the arterioles, we see a massive drop in velocity, and then we have a very slow velocity in the capillaries. The reason that is important is it enables time for exchange of materials. Blood is travelling at really high velocity in the arteries because it has just been pumped. It has just been pumped by the left ventricle out. It is under really high pressure, and that's what we see here: really high pressure, and then the pressure drops in the arterioles, and then the pressure is really low in the capillaries, and the pressure remains low in the venules and in the veins. Whereas if we look at the velocity increases from the capillaries into the venules and the veins. So there's something other than pressure affecting the speed. And we'll talk about that in a bit. Let's look at this one, total area. So total area, if we think even in the largest arterioles, if we think in the aorta, the area, and this is specifically referring to the area of the lumen, so the space in the middle, the area is really small because there's only one of them. But as it branches out into more and more of them, this is additive, okay? So we're adding all these lumens from the smaller vessels together. And so even though each individual one might have a small area, if you add them all together, the total area is very, very large. So at first, the total area is low. And then as we move into arterioles, we start to get the branching. And so it goes up and up and up, and then obviously we have loads of branching in the capillaries, massive. Venules, they all start to join back together again. So we get smaller and then smaller and smaller in the veins. Just looking a second at the units, mmHg means millimeters of mercury. That is what that is measured in. So if you cut an artery, blood spurts out because it is at high pressure and it is at high velocity. Whereas if you cut a vein, blood sort of meanders slowly out of your body because it is travelling at low pressure and pretty low velocity. So let's take a minute to have a look at this graph here. It's essentially showing us the same thing as below, but I quite like the way that it's displayed. So we've got blood velocity in centimetres per second, and we've got a cross-sectional area in centimetres squared on the graph. So we can see that as blood travels through the aorta and then the arteries and into the capillaries, the velocity drops as it goes into the arterioles, very low in the capillaries, and then increases again. Whereas in terms of cross-sectional area, it's low, it rises gradually, then it has a steep rise at the venules and the capillaries, and then a steep drop down into the veins. And really the question we have to ask ourselves is, why does the velocity increase again? There must be some other force acting. Well, the main reason that blood flows through the veins is, there's no force from the heart. Like, it's not blood pressure, it's not the pumping of the blood. That's what gets blood through the arteries. Blood primarily moves through the veins by the rhythmical contraction of a smooth muscle in the vessel wall. So the smooth muscle in the veins is contracting and pushing blood along. And B, by the action of skeletal muscle. So as you move your body, tiny movements of your skeletal muscle, these push blood along in the veins. This is what keeps blood moving along in the veins. This is like the main way we get blood along. Because most veins are moving blood against the pull of gravity. Think about it like, you've just pumped blood all the way down to your feet. You've got to get that blood all the way back up from your big toe all the way back up to your heart. That's quite an ask. So you've got the skeletal muscle, the contraction, you've got the little smooth muscle contractions, and then you've also got the really important job of the valve. These one-way valves prevent backflow. This is really important. So skeletal muscle contraction aids in venous blood flow, and this means that it's really important to always keep moving. This is why, like, if you sit down or you lie down for a really long time, it basically causes blood to pool in the extremities of your body. So you've got to move around, like you've got to stay active. This is why you start to feel a bit funny if you sit down for too long or if you lie down for too long. Just gone on a slight tangent here. Have any of you ever heard of something called varicose veins? Varicose veins are veins that become enlarged because there's something wrong with the valves and they no longer close properly, and this allows blood to flow backwards in the vein. So varicose veins are often most prominent on your legs. This is because your valves are working hardest in your legs because they're pushing, the blood is moving, not pushing, I shouldn't have said that, the blood is moving most against gravity, and so the valves are having to handle way more backflow, blood coming down and hitting them way more regularly in the legs than they would in any other part of your body. So that's why you're most likely to get varicose veins there. So let's just take a minute to confirm we understand the jobs of each of the different parts. So we've got this smooth muscle that can contract and relax to control the flow of blood, and we talked about the importance of that in the veins. We've got the elastic layer that helps to maintain a constant blood pressure by stretch and recoil. We've got the endothelium, the inner layer. The endothelium is smooth. This reduces friction. This is really important because friction resists blood flow. The more friction there is, the slower the blood will flow. And they're thin to allow diffusion. Now this is really only valid in capillaries. Capillaries only have endothelium, okay? They're one layer thick, and that's where we need diffusion to be occurring. The lumen is the central cavity of the blood vessels through which the blood flows, and you've got this connective tissue which resists pressure changes from both within and outside. So we've covered quite a lot of information in this video. We've covered, generally, why organisms need a circulatory system. We have covered the difference between open and closed circulatory systems. We have looked at the difference between single and double circulatory systems. We've looked at the difference between the cardiovascular system and the lymphatic system. We've talked about our history of our knowledge of the circulatory system, and we talked about Galen, who did a lot of early work on the circulatory system, and then we talked about William Harvey and how he developed our understanding of the circulatory system. We talked about the structures and functions of arteries, veins, and capillaries. We talked about the difference in total cross-sectional area between arteries, veins, and capillaries, the difference in the velocity of flow of blood through them, and the difference in blood pressure. We talked about what each one is made up of, so the different layers of arteries, veins, and capillaries, and what the job is of each of those parts. You've been introduced to lots of key terms today, and I hope that you have found it useful. We will carry on talking about this soon. Have a lovely day.